applications. So as you might have seen that while we were discussing about this chapter, we have discussed about the different types of applications of the biotechnology, whether it is a application in the agricultural field or the medical biotechnology, or whether it is related to diagnostics. So in today's lecture, we are going to discuss about the new topic, and the new topic is transgenic animals. So what is mean by the transgenic animals? So transgenic animals are the animals which are actually going to carry the gene from the foreign organisms. What is mean by this is that you are actually going to have the transgenic animals where you are actually going to alter the genome of these organisms in such a way that it is going to be heritable manner, and if you want to do that, which means if you want to alter the genome of an organism so that it is going to be heritable, then you have to introduce the gene or the foreign DNA into the genome of its zygote or the embryo, so that when the zygote or the embryo is going to develop into the full organisms, all the cells like somatic cell as well as the genetic cells are going to have the genome of the same type, and these genomes are actually going to carry this foreign DNA. So in this case, the foreign DNA what you are going to use to introduce into the organism is called as the transgenes, because the gene is going to come from the separate species, and the process through which you are actually going to introduce this particular gene into the animal, and you are going to prepare the transgenic animal. So the overall process of preparing the transgenic animal is called as the transgenesis. So there are three ways in which you can be able to alter the genome of an organisms. Let's discuss about that. So you have sexual reproduction, which is a natural mechanism through which you can be able to alter the genome of a particular organism. In the sexual reproduction, what you are doing is you are taking a male, which is the blue color circles, and then you are taking a female, which is going to be of different characters like the red balls. And then when they are going with the sexual reproductions, they are actually producing a progeny which is actually going to have the mixed characters. So here the mixed characters are actually going to have the genomic content, 50% genomic content from the male and the 50% genomic content from the female. And because of that, this progeny is going to be called as the hybrid varieties. These progenies will not called as the transgenic animals, because the genes what you are introducing into this particular organisms are coming from the similar kind of species. For example, if this is a rabbit, this is also a rabbit, but this could be a rabbit with the some definite phenotype. So because of that, the progeny what you are going to get is actually going to have the properties from the male as well as the female, and that's how this progeny is going to be of a improved varieties or improved characteristics. But these progeny will not going to carry any gene from the trans species. It is going to contain all the gene from the cis species, which means it is going to contain the DNA from the same species, and that's why these kind of hybrids are not called as the transgenic animals. Compared to that, you can be able to generate a transgenic animal with the help of the genetic engineering. If you recall, while we were discussing about the biotechnology principle and processes, we have discussed about how you can be able to generate the chimeric DNA. So what you have to do is you have to first isolate a genome, and then from the genome you have to isolate the gene of your interest, and then the gene of interest has to be digested with the restriction enzymes. Similarly, you have to process the vectors, and then you are going to put them together for the ligation reaction, and that's how you are actually going to get the chimeric DNA. This chimeric DNA then has to be introduced into the organism of your choice, and now at this stage this particular DNA either can remain within the cytosol. For example, in this case, when we are introducing this into the bacteria, it is actually going to give you the transformed bacteria. But there are other cases. So this particular type of bacteria will not be called, so it's going to have carry the gene from the foreign organisms, but it is not going to be a permanent species, because after the few cycles this plasmid is going to be removed, for example. And the other case could be that you actually going to clone the DNA into a vector, then you introduce this vector DNA into the organisms. Once the DNA will enter into the nucleus, so at this stage, once you enter the DNA into the organism, it could have two choices. One, it is actually can go into the cytosol, like uh, it is actually can go into the cytosol, or it can go into the nucleus. So if it is goes into the cytosol, it will remain there and it will express for some time, but afterwards it will not going to integrate into the genome, because and it will be going to be removed. So these kind of organism where you are going to